Human Embryology, Part 2. Lecture Plan. Neurulation, Histogenesis, Extraembryonic Organs. In previous lesson, we spoke about structure of germ cells, stage and process of embryogenesis, especially about fertilization, cleavage, gastrulation. As a result of gastrulation, 3 laminar embryo was formed. Embryo after gastrulation have a 3-layer: ectoderm, entoderm, and mesoderm. The next stage have process development of every layers, development of ectoderm, development of entoderm, and development of mesoderm. At different tissues and different system of organs will develop from these 3 layers. So, differentiation of ectoderm, entoderm, and mesoderm. Histogenesis. This is process differentiation of mesodermal, entodermal, and ectodermal germ layers. Differentiation of mesodermal germ layers. There are 3 parts of mesodermal germ layer: paraxial part, intermediate part, and ventral part. Paraxial mesoderm in this side, and this side. Paraxial mesoderm is organized into segments, somites, by the beginning of the 3rd week. Each somites consists of mesodermal cells, which become sclerotome, myotome, and dermatome. Sclerotome will produce bone tissues, and especially bone tissues of vertebra. Myotome will produce muscle tissues, especially skeletal muscle of body and hand and legs. Dermatome will produce derma of skin. Then, intermediate mesoderm, this part, violet color in these drawings and in this side of drawings, intermediate mesoderm, which temporarily connects paraxial mesoderm with the lateral plate, differentiates into the urogenital structures, future nephratomes, or the nephrogenic cord. So this part, intermediate mesoderm, have a name, or will develop 2 nephratomes, or maybe nephrogenic cord. This part produce kidney. We have several types of kidney: embryonic kidney and definitively kidney. All these kidney will form from nephratomes in this part, in intermediate part of mesoderm. Then, the ventral unsegmented mesoderm, splanchnatom. The ventral unsegmented mesoderm, splanchnatom, is divided into 2 sheets, the visceral and the parietal part. Both of these leaves will form mesothelium. In this diagram you can see, especially in this diagram you can see splanchnatom, ventral unsegmented mesoderm. This part visceral sheet of splanchnatom and parietal sheet of splanchnatom. They form cavity inside. This cavity inside have a name coelom, intraembryonic coelom. Visceral sheet also forms myoepicardium plate, which will form the myocardium and epicardium of heart. In this place, in cranial part of embryo, visceral sheet will form myocardium and epicardium of heart. Right and left side of heart, these two part unit together and forms the whole heart. So, differentiation of mesodermal gem layer inside. The mesodermal gem layer divide in three parts: paraxial part, intermediate part, and ventral part. Paraxial part forms somites, segmented structures in paraxial part of mesoderm. Cells of somite divide and forms three groups of cells: sclerotome, myotome, and dermatome. Intermediate mesoderm, intermediate part of mesoderm, produce nephratomes or nephrogenic cord. This is future kidney, embryonic kidney, and definitively kidney. Ventral mesoderm forms cavity, interembryonic xylem, and this cavity have two sides or two sheet, parietal sheet and visceral sheet. Parietal sheet and visceral sheet produce mesothelium, special squamous epithelium type, and in this part, in cranial part, will produce myocardium and epicardium of heart. Xylem in embryo in future will forms cavities in body. I mean lung cavity, thoracic cavity, and abdominal cavity. Both these cavities will covered with lining by mesothelium. Mesothelium was formed by visceral and parietal unsegmented mesoderm. Differentiation of endodermal gem layer, or derivatives of endodermal gem layer. So, endoderm forms yolk sac and forms lower part of embryo. This side. Endoderm, this endoderm forms lower part, lower layer of embryo, and then this side form yolk sac. These gem layers, endodermal gem layer, covering the ventral surface of the embryo and forms the roof of the yolk sac. Then it is folding. They are most pronounced in the region of head and tail of embryo. As a result, cephalocaudal folding, a significant part of the endoderm lined cavity, is included in the body of the embryo proper. In front, the endoderm forms the foregut. In the tail region, it forms the hindgut. The part between foregut and the hindgut is the midgut. The midgut temporarily communicates with the yolk sac by the way of broad stalk of the vitelline duct. The allantois is separated. By the fifth week, the yolk sac duct, allantois, and umbilical cord vessels are limited to the area of the umbilical cord. So this part is head of embryo, this part is tail of embryo. And yellow color, this is endoderm. Amniotic cavity, this part, upper amniotic cavity located in upper part above embryo, and amniotic cavity forms folds from cephalic region and from caudal region, from head of embryo and tail of embryo, and divide endoderm lining tissue, endoderm layer, in two parts. This part become yolk sac endoderm. This part become embryonic part of endoderm, and inside embryo, this part of endoderm forms a gut, primary gut, foregut, hindgut, foregut, hindgut, and midgut. This primary gut, this is future gastrointestinal tract. Then folds is dipping and forms vitelline duct or broad stalk, this or this, and vitelline duct. Vitelline duct, this is tube which connects primary gut with yolk sac. Yolk sac will disappear in the 3rd month of embryonic development, because in human yolk sac lost their first function, nutrition function, because embryo receive nutritive substances from placenta. So the main process in differentiation of endodermal germ layer, this is process of form primary gut, because primary gut, this is future gastrointestinal tract. Then and differentiation of ectoderm. Differentiation of ectoderm. This is part of histogenesis also. So histogenesis, this is process of differentiation of 3rd layers, ectoderm, endoderm, and mesoderm. Mesoderm divide in 3 parts of mesoderm and forms notochord, forms somites, forms coelom, forms heart, forms nephrogenic cord and embryonic kidney. Endoderm, differentiation of endoderm produce primary gut and gastrointestinal tract. And ectoderm, differentiation of ectoderm produce nerve system. The process of nerve system formation is neurulation. The process of nerve system formation is neurulation. Neurulation, the process of forming nervous tube, which start from 18th day. Part of ectoderm above the notochord becomes a neural plot. In this diagram, you can see ectoderm, mesoderm red color, and endoderm yellow color. In middle part of mesoderm, you can see round shape structure. This is notochord. In this diagram, you can see notochord and ectoderm and mesoderm, somites of mesoderm. So, part of the ectoderm above the notochord, become a neural plot, like this neural plot and like this neural plot. The lateral edge of neural plot become more elevated and form neural folds, become more elevated and form a neural folds. And the mid-region forms a neural groove, and mid-region forms the neural groove. Then, gradually, the neural folds approach each other in midline and then fuse. Fusion begins in the cervical region, in this part, in these diagrams, in cervical region and proceeds cranially and caudally. This is diagrams. This is diagrams, not transverse section. It diagrams if you see at embryo above. You can see in this part folds, neural folds, and fusing in neural folds. This neural plate and this cranial part, this caudal part, this cervical region and fusing start in cervical region, proceed in cranial and caudal directions. As a result, a neural tube is formed. Until fusion is complete, the cephalic and caudal ends of neural tube communicate with amniotic cavity via the cranial and caudal neuropores, respectively. Closure of the cranial neuropore occurs at approximately day 25, whereas the posterior neuropore closes at day 27. The neurulation is then complete and the central nervous system is represented by a closed tubular structure with a narrow caudal portion of the spinal cord and a much broader cephalic portion characterized by a number of dilatation in the brain vesicles. As the neural folds elevate and fuse, cells of the lateral parts border or which have neural crests, this part and this part, neural crest of the neuroectoderm begins to dissociate from their neighbors. This cell population, the neural crests, will migrate and give rise parasympathetic and sympathetic ganglia, dermal melanocytes, adrenal medulla, organs of senses, and spinal ganglion. So, neural tube in future will form spine cord and brain. Neural crest, and also neural cells will form ganglia, nerves, dermal melanocytes, and organ of senses. Extraembryonic organs. Extraembryonic organs. As mentioned before, in the course of early-stage human embryogenesis, some temporary organs appear to supply the normal life growth and development of proper embryo. They are so-called extraembryonic or provisional organs. Initially, yolk sac, like a provisional organs, arises in fishes. Then amnion, serous, tunic, allantois, chorion, placenta, and umbilical cord were developed in animals. There are such human extraembryonic provisional organs as amnion, allantois, chorion, placenta, and umbilical cord. In human extraembryonic provisional organs, we cannot find serous tunic. So, amnion. Amnion is a fetal membrane which begin its development at the third week of embryogenesis when embryoblast delamination produce epiblast and hypoblast formation. Amniotic cavity appear in the epiblast, then extraembryonic mesoderm visceral sheet cover the structure. Thus, amnion consists of epithelium of ectodermal origin and mesodermal connective tissue. Amnion has protective function, homeostatic function, and produces amniotic fluid. Amnion contain amniotic fluid, which epithelium of which was produced by epithelium of amnion. Amniotic fluid need for development of embryo because embryo can develop it only in water environment. This water environment, amniotic fluid contain a lot of water. This water environment protect embryo against mechanical damage. Protect embryo also against increase or decrease temperature. The yolk sac. The sac appeared from the hypoblast. In human embryogenesis, yolk sac had lost its first and principal function, to store the nutritive substances. But it has some more important function. For example, it performs hematopoietic function. It is the source of henocytoblast and primary vessels appear in its wall. The wall of yolk sac consists also of endodermal epithelium and mesodermal connective tissue. Exactly, endodermal epithelium produce gonocytoblasts. This is primary reproductive cells they need for development reproductive system. And mesoderm produce primary vessels. This is start of development cardiovascular system vessels. Also, this mesoderm produce the first blood cells. Allantois, like a finger-like process of primary intestine ventral wall, it appears at about 16th day of development. It grows up from the caudal portion of intestine into the connecting stalk, amniotic foot, and may be absorbed there till 2 months. Allantois consist of cuboidal epithelium surrounded with connective tissue. It is excretory, breathing function. It is the guide of blood vessels in umbilical cord, and at last, it performs immune function. Primary B lymphocyte appears in its wall. Umbilical cord. It is tube which arise of connecting stalk, amniotic foot, when the amnion is growing. That's why it is covered with the cuboidal epithelial of amniotic origin and it's filled up with Wharton's jelly. Wharton's jelly is connective tissue with special properties which contain much glycosaminoglycans and numerous macrophages. There are 2 arteries and 1 veins and the remnants of yolk sac and allantois in the umbilical cord. It performs function of excretion, breathing, and nutrition. Chorion is developed from trophoblast when it is differentiating into cytotrophoblast and syncytotrophoblast, and underlying with extraembryonic mesoderm. There are two periods of chorion development, pre-villose stage and villose stage. Pre-villose till 7-8th day, and a period of villi formation or villose stage from 9 to 15th day. There are several types of villi. There are primary villi, there is secondary villi, and there are tertiary villi. Primary villi appear when the cells of the cytotrophoblast proliferate locally and penetrate into the syncytium, thus forming cellular columns surrounded by syncytium. Secondary villi appear when mesodermal cells penetrate the core of primary villi and grow in the direction of the uterine mucosa. They contain connective tissue and a layer of cytotrophoblast cells and layer of syncytium. And tertiary villi, by the end of the third week of mesodermal cells, the core of the villus begin to differentiate into blood cells and small blood vessels, thus forming the villous capillary system. The villi are now known as the tertiary stem villi, and during the fourth week of development, this type of villi can be found over the interior surface of chorion. So, there are differences between primary, secondary, and tertiary villi. More early villi is primary villi. They contained only cytotrophoblast and syncytium trophoblast. Secondary villi contained cytotrophoblast, syncytium trophoblast, and mesoderm, which produce connective tissue, loose connective tissue. Tertiary villi, this is more oldest villi because they have not only syncytium trophoblast, cytotrophoblast, and also they contain loose connective tissue, mesodermal, and also they contained blood vessels. Placenta, the next extraembryonic organs. Placenta is a special organ which consists of two principally different structures of both maternal and fetal. And also placenta consist of chorion with their villi and decidua basalis. Decidua basalis, this is tunica mucosa of uterine. This is maternal part, maternal tissues with maternal DNA. So, placenta, this is a special organ which consists of two parts, maternal part and fetal part. Fetal part, this is chorion with their villi. Maternal part, this is mucosa of uterus. There are different types of placenta in mammalian. In human placenta is of discoidal type and gemahorial. It means that the placenta have a discoid shape, not oval shape, only discoid shape. And gemachorial, it means chorionic villi receive nutrition from maternal blood. So, human placenta is discoidal type and gemachorial type. Discoid, it means they have a different, they have a discoid shape, and gemachorial, it means the chorionic villi receive nutrition from maternal blood from maternal part. The fetal part of placenta is made up of a chorionic plate with its villi. And cytotrophoblast layer, which covered the villi, and the intervillous space. The chorionic plate consists of amnion, it means covered by amnion. The X covered by amnion in this side, which which contacts with amnion, with amniotic fluid and embryo. In another side, which contacts with the maternal body, maternal tissues, is chorion covered by cytotrophoblasts. The chorionic plate consists of amnion, the extraembryonic mesenchyme, the cytotrophoblast, and cytotrophoblast. Cotyledon, this is a structural unit of placenta. It is a willow tree growing from chorionic plate, located intervillous blood vessels. Each willow tree consists of stem villi, anterior villi, terminal villi. Each villi contain vessels with fetal blood, extraembryonic mesenchyme, trophoblast, and cytotrophoblast. The maternal part is made up of the maternal tissue, Decidua basalis. The Decidua basalis consists of decidual cells. It is large cells with abundant amounts of lipids and glycogen. During the 4th and 5th month, the decidua forms a number of septa, the decidual septa, which project into the intervillous space but don't contact with the chorionic plate. As a result, in this septum formation, the placenta is divided into a number of compartments with cotyledons. In the second half of pregnancy, lacuna surface is covered by special proteins membrane, or fibrinoid. Similar fibrinoid of Langhans covers chorionic villi, similar fibrinoid of Nitabuch covers chorionic plate. And the main functions of placenta are the next. The nutritive function. Maternal blood in placenta brings nutrients for the fetus blood. Excretory function. Maternal blood takes waste products and metabolites from fetus blood. Protective function. Immunocytes of maternal blood protects fetus against viruses, bacteria, and other infections. Respiratory. Maternal blood and placenta brings the oxygen to the fetus blood and takes carbon or dioxygen from the fetal blood. Endocrine function. In humans, the placenta also secretes hormones, human chorionic gonadotropin, human placenta lactogen, estrogen, and progesterone. They are important for development of pregnancy. So, this is umbilical cord to embryo. This is amniotic cavity in this side, amniotic cavity, and umbilical cord covered by a layer of amnion, and this part, chorionic plate, also covered by amnion. From this to this is chorionic plate. This chorionic plate in this side covered by amniotic wall, amniotic tissue. Amniotic tissue, this is amniotic epithelium, simple cuboidal epithelium, and mesoderm, which forms connective tissue. Chorionic plate also contains connective tissue. In this side, they have cytotrophoblast and syncytium trophoblast. Then, in connective tissue, we can find a lot of vessels. Which were very branched. Then, chorionic plate have a lot of villi. In human placenta, there are about 200 villi. This one willow tree have a name, cotyledon. Cotyledon, this is functional unit, structural functional unit of placenta. There are about 200 of cotyledon in human placenta. Cotyledon, this is tertiary villi. In these diagrams, this tertiary villi, maternal villi, with vessels, with connective tissue. And we have a name for a different part of this cotyledon. This part, main part, have a name, stem villi. Stem villi, like stem in tree. This part, anchor villi, we have this part, we have anchor. We name it anchor villi because they, like anchor, contacts with the maternal part. Then very little and very thin willow. They have name terminal villi. These terminal villi have very thin wall and a lot of capillary, and they need for exchange gases, exchange nutritive substances, exchange waste subproducts of metabolism between this blood and between this blood. In this site, in this place, have a name lacuna. This is place between villi, between maternal part, between septa, between chorionic plate, and in this part you can see maternal blood. All these lacunas was filled by maternal blood. So maternal blood have open circulation. Fetal blood have closed circulation. What does it mean, open and closed? Closed circulation when blood can circulate only inside vessels. This is blood of fetus, only inside vessels. This blood, maternal, this open circulation because blood flow in this space and then arteries. From arteries opens and blood flow in this place, and then they return to veins, maternal veins. But maternal blood and fetus blood never contacts because they maybe have a different group of blood. They have especially they have a different DNA. Therefore, maternal blood and fetus blood never contacts. These bloods can only exchange some substances through the wall of villi, through the connective tissue, through the syncytium, cytotrophoblasts, which covered all villis, and also above the syncytium in late stage of placental development, we can see fibrinoids. Fibrinoids, this is layer of proteins. It also need for protect fetus tissues against maternal immune cells. Then this part with willow tree with cotyledons, this part is fetal part. This part is maternal part. Maternal part, this is wall of uterine, inner part wall of uterine, mucus of uterine. This mucus change and become decidual basalis. This is myometrium, this is endometrium, and endometrium or another name, muco layer, mucous tunica mucosa in uterus become decidua basalis. Decidua basalis contain connective tissue, contain epithelium, and contain vessels, and also contain decidual cells. Decidual cells were large cells which have a lot of nutritive substances like a glycogen. It need for nutrition of embryo, especially the first 3 months when placenta grows. So this part is maternal part, and they have a maternal tissues with maternal DNA. This part is a fetal part, which have a fetal tissues with fetal DNA. In microphoto, you can see chorionic plate, and this line amnion. Amnion have a two color, more violet color, amniotic epithelium, and this color, pink color, amniotic connective tissue. This is chorionic plate, connective tissue of chorionic plate, and in this part, violet line, syncytiotrophoblast and syncytiotrophoblast or syncytium and cytotrophoblast, not epithelium. Then you can see stem villi, like this, stem villi. And in this part, you can see parts of willow, a lot of different type of willow. Terminal villi, very large branch of willow tree, and anchor villi, more very little. Very little part of willow tree, and anchor villi, large part of willow tree. And you can see red color. This is maternal blood. Maternal blood flows around all villi. Flows, and they flow in lacuna. It also fetal side and in this side maternal side, amnion epithelium and connective tissue. Then chorionic plate, which contain a lot of blood, fetal blood vessels, and stem villi, like this stem villi, with vessels also. And you can see terminal villi, large branch of cotyledon of willow tree. So terminal villi, microphoto of terminal villi. All terminal villi contain connective tissue, contain vessels, this is vessels, and contain syncytio and cytotrophoblast. And this is lacuna. Lacuna contain blood of maternal blood. Erythrocyte is red color. This is erythrocyte of maternal blood. That's it, the endometrium. Myometrium with vessels, endometrium which become the pseudobasalis, trophoblast, placental anchoring villi. Anchoring villi have closely contact with maternal part. So there are gemahorial or placental barrier. It is dividing membrane between a maternal and fetal circulation. There are maternal circulation, maternal blood, and fetal blood. Between them, we have tissues. We have connective tissue of willow. We have syncytium trophoblast, cytotrophoblast of willow, and also in diagrams, in regal microphoto, we can see fibrinoid of Langhans. This is special proteins of blood which covered all willow and protect willow against maybe immune cells, maybe in maternal blood, maybe also against viruses, bacteria. So gemahoria barrier or placental barrier, this is barrier between maternal blood and fetal blood. It's very important. Barrier is very important to safely protect embryo from different negative influence from bacteria, viruses, infection, and also protect against immune cells of maternal blood.